Hello, everybody. Welcome to yet another Journal Club brought to you by Life Extension Advocacy Foundation, LEAF. I'm Dr. Oliver Medvedik, coming to you from New York City from my bedroom. So hopefully I'll be back at the Kambar Center at Cooper Union, where I normally used to do this before the pandemic. And we're slowly opening things up here in New York City. Workers are trickling in, social distancing is being practiced. So I hope everybody is safe and sound wherever you're at. So those of you who were able to join us during our virtual conference last week, Thursday and Friday, thanks for coming. Thanks for attending. I was emceeing the event. There was a lot of participants at the event, a lot of great speakers. A lot of that is, well, I think a lot of that is online right now. Those of you who had VIP access, I think you can have access to everything that was on there already. Some of the other stuff is going to be trickling in, but I think we're all proud of the fact that we were able to pull off this virtual conference with just a few little hiccups, but I think it went pretty darn well. It was a lot of solid, up-to-date information, and hopefully next year, 2021, we will be doing a live conference, as we did for our first two conferences, for ending age-related diseases. So stay tuned for that. But right now, we just finished one up, so we're going to be taking a little breather. DDA. I paid also to support the event, but I had no time to listen yet. Well, all that's online, so dive in. We've got all the all the talks there, and as I was emceeing and kind of keeping an eye on the clock and doing other things, I kind of had only one ear on the speakers, so I'm going to have to review some of the talks as well. The ones I did manage to listen to were fantastic. So we have a lot of great resources that are available to you, so do check that out on our lifespan.io website. So back to our journal club. We are doing our, I don't remember what number this is for this journal club. So Steve just mentioned talks will be online for 2 weeks via the event platform. Then we'll be putting all the content, I'm going to cut off somewhere. Okay. Anyway, so back to the journal club. So we have a paper from the Blasco lab. So this is a telomerase paper, and way back a little while ago, we talked about a condition that's a manifestation of a chronic disease basically related to aging, idiopathic pulmonary fibrosis. I believe one of the papers we did was from the Mayo Clinic from James Kirkland's lab, who was a speaker at our event. And they used a different methodology. I think they used senolytics, and this was a small scale trial. So this was actually already getting into clinical trials. This paper here that we're going to be covering is a telomerase paper. And let me just pull that up, share my screen. Okay, pull this up. Hopefully this is still lined up fairly well, I think. So this is from Pinheiro, Hermida et al. Blasco lab. Its telomerase treatment prevents lung profibrotic pathologies associated with physiological aging, i.e. idiopathic pulmonary fibrosis. So this is a lung condition where essentially the active cells in your lungs that basically create surfactant, which keep your lungs open and allow the alveoli, which are the lung sacs, to basically open up and allow gas exchange to occur. You basically have scarring taking place. You have fibroblasts end up dividing instead, and basically you lose the ability. Your lungs basically can't basically do adequate gas exchange. So it's a disease that afflicts humans, and it's very it's a lot of suffering, and the fatality rate is pretty high if you're diagnosed with it. So any treatment for it is would be very welcome. Here, what they did was they demonstrated that adding in telomerase or extra telomerase, and they use an adenoviral vector, AAV, to basically add in, I believe they added in TERT, which is catalytic subunit telomerase, to mice. So this is a mouse study using C57 black 6 mice. And they use two types of mouse models here. So I'm going to stop sharing here. So they use two types of mouse models here. They basically used mice that were normally aging. So, you know, very, very old mice, many weeks old. So that's good. So basically normal aging where the mice were manifesting, IDF. And also mice that were kind of an accelerated aging phenotype that were telomerase negative mice that were multi-generational. So mice have very, very long telomeres. So a lot of these, some of these manifestations of very short telomeres happen only like after or passage them. So these are like, I believe, generation 3 mice that were knocked out for telomerase, right? So they had these two models. So one is kind of more realistic aged mice, and the other one is more hyper-accelerated telomere attrition mice, right? And basically the punchline is that these effects, by adding in telomerase, meaning a reversal of the consequences of IDF, idiopathic pulmonary fibrosis, these effects were reversed in both mouse models, which is extremely promising. So, you know, telomerase, you know, is one of the, or shortened telomeres is, you know, one of the, you know, supposed hallmarks of aging that's found in many organisms and found in actively dividing cells. And these are the types of cells that we see in the epithelia of lungs, which are actively dividing cells. So the hypothesis would be that if we basically rescue telomerase and rescue, you know, telomere extension, that perhaps we can basically rescue, you know, this disease state and bring the cells back to normal. So that is what this paper actually suggests using a mouse model. So let's go back. So this bodes well for potential human trials for administering not just senolytics, as was done in, I believe, in the Mayo Clinic paper that we covered in a very, very, very small scale clinical trial, but also other using other hallmarks of aging, such as adding back telomerase as a potential therapeutic. So that's pretty good. So let's go back to sharing the screen here. Okay, so what did they exactly do? So there's like 9 figures in this paper, lots of bits and pieces to all these figures. I probably won't be able to go through all of them, but let's do the best that we can. So this, the first kind of half of the paper kind of sets up sort of the experiment and how they performed it, and kind of the expectations you would see. So up here in the upper right-hand corner, you have your mice, heterozygote mice that were knocked out for telomerase cross, so you can get telomerase, wild type mice, heterozygotes, and then G1 TERT minus mice. So these are the mice that are completely knocked out for telomerase, and then G3 TERT minus mice, which are basically after, it's a 3rd generation mice. So they have very, very short telomeres. They don't live a very long time. So this is kind of like hyper accelerated telomere attrition, and let's just say accelerated aging, if you will, from a telomere standpoint. And this is basically their experiment. So mice, going from 0 weeks to very old mice, 125 weeks, and then everything in between, where they sacrifice the mice and collect lungs and basically do all of these experiments on the mice. Let's see, I'm going to go to the chat here briefly. Okay, it's just technical questions. People are asking about chatting. So do you ask questions here? I'm not sure if I'll have the answers, but we'll try to do the best that we can. Okay, So they looked at two different types of cells here, these AT2 cells and these club cells. So these are alveolar cells that basically produce surfactant. They're epithelial cells that, are required for basically proper lung functioning, right? And basically what we have here is a slide looking at immunofluorescence, so antibody staining. And what we're looking at here, I believe, all of these boxes, you have blue DAPI nuclei that are stained. Those little red speckles that you see are telomeres. And then you have all these green speckles, which are actually the AT2 cells, and then the club cells that they're looking at. And I believe this is another form of staining that we have on the side to side, which is this SFTPC and SGB1A1. So this is the type of marker that's specific for these cells. And then you have mice in various ages. So 5 weeks, very young mice. So we're looking at very young mice that are normal, wild type. That's the TERT++, upper right, left-hand corner. And then we have the G3 TERT minus. And so you can kind of consider that to be a positive control, because these mice have very short telomeres. And if we zoom in, kind of, being a bit qualitative here, you can see that just by eye, without doing any kind of statistics, you can see that there's much less telomere foci or speckles here on the left. And then the same thing, basically for 8 to 9 weeks, 42 to 49 weeks, and then 92 to 125 weeks. And then the really old mice, they don't have G3 TERT minus mice because they're all dead by that point. So you basically have your wild type aged mice. So what they want to do here is, well, as the figure legend shows, telomerase deficiency and natural aging leads to telomere shortening and the reduction in the number of AT2 and club cells. So basically here they show if not a causation, a correlation between short telomeres or shortening telomeres and a reduction in the number in their field of view of these cells that are required to maintain basically proper lung function. And the numbers are on the bar graphs, the histograms are on the right. So I'm just gonna take a look at one of them. Just move my box here out of the way so I can see it. Let's take a look here. So for example, in the top one, which is the mean telomere spot intensity in SFTPC AT2 cells, the gray is G3 TERT, and the white box is TERT++. So these are wild type cells, and these are the ages. So 5 weeks to 92 to 125 weeks. And basically, you can see that, they're fairly higher than, your G3 TERT, your telomere spot intensity, but the number goes down. Even in the aged population, not as dramatic, not as low as obviously telomeres minus cells. And finally, N is 0 because you don't have mice for your G3 TERT minus because they're pretty bad off. They're dead. So okay. So basically, I'm going to take a little breather here if anybody has any questions. We're going to pop back into the next figure. But basically, these first several figures are just basically setting up what's going wrong with the mice, essentially aged mice and kind of hyper-accelerated telomere attrition mice, and kind of show that for what they're looking at, the problems are pretty similar. So there's, so they're showing you that there's this correlation between old age and telomere attrition. Although I don't think they actually measure telomere length per se. They sort of do with the immunofluorescence and the kind of the foci, you have less of them. Okay, so let's go back to share the screen. Okay. So now what we're looking at is a different type of stain. And I mentioned before that, you've got these normal cells that are basically responsible for your alveolar function, right? So alveoli, as you remember from Bio 101, they're all 102. You basically have their sacs that basically allow gas exchange to occur between capillaries. And, the cells there is highly specialized to produce a whole bunch of things like certain surfactants, which are things that basically keep these little sacs from collapsing and sticking to one another. Otherwise, you'll have a real big problem for increasing the surface area so you can get proper gas exchange. And one of the problems with, sorry about that. That was mouse malfunction, just went out of control. One of the problems with aging is that you have less and less of these cells as you get older, and you have other things happening which don't necessarily improve the functionality of lungs, such as the buildup of cells that are fibroblasts that are not specific for this functionality, and they produce other things. So here we have staining of, so just going to read the legend here. So premature aging by accelerated telomere shortening and natural aging in the mouse lung result in increased fibroblast presence and activation and collagen deposition. So basically we have markers for fibroblasts such as vimentin. We have various markers such as SMA, Sirius red, and Masson's trichome staining, which basically stains for collagen, which is this kind of bluish ring that you see here. And basically, if you get more and more of these fibroblasts and more of this collagen that is appearing, that is inversely correlated with lung function. So more of that is bad, right? So the same kind of table that you see here, the same setup in this. So this is, staining in microscopic cross sections. Little. You can see it's little bar tells you it's 20 microns. And then you have again 5-week-old mice, 8 to 9 weeks, 42 to 49, and 92 to 125. Comparison between and the number of mice, 4 animals per group. So alveolar and bronchi cross sections. So you have your wild type mice, which are TERT ++, which were derived from the same C57 black 6 background as the G3 TERT -— mice. So that's always important, right? You don't want to do apples and oranges comparisons. So that's very good. They had mice that were from the same batch, right? So they had heterozygote mice that were knocked out for telomerase. The methodology is in the materials and methods. And they crossed the mice, and they basically, basic genetics, Mendelian genetics, and you get your TERT -, TERT ++, and TERT ± heterozygote mice. And then they, from that same litter, they basically then propagated mice that were G3 TERT -. So those, again, are your kind of accelerated telomere attrition mice, which are, I would say, call that like a positive control. And then you have your normal mice, which are allowed to age right to a old age of 125 weeks, right? So all of this data here on the left is then converted into histograms on the right, and basically just through scoring. So whatever methodology we used, is presumably unbiased. And again, so the Vimentin area over HPF, that's I believe that's high. I forgot it's basically your field of view in a microscope, right? During high magnification. I think it might be 400x, but basically it's the amount of Vimentin area that's stained, which is, indicative of fibroblasts in the field of view that they're measuring in a microscope, right? And you can see just by the histogram that after a period of time, this goes up, right? For both, this is n = 4 for both mice that are knocked out for telomeres and also your normally aged mice. So it's not a huge surprise that you have aged lungs, and they're basically exhibiting aspects of the aging process here by having more cells that are producing extracellular matrix, right? So I'm just not going to go through all of these here, but they're looking at, for example, D and E, alveolar collagen per same field of view, and then peribronchial collagen. So they're looking at it in alveoli and also the collagen, increase in bronchi, which are basically the little tubes that feed into the alveoli, and you have this increase as well. So I'm going to go into the chat here. Yeah. So they're establishing that that you're getting these effects in both of these mice. Both mice meaning the accelerated aging, and when I say accelerated aging, I really mean accelerated telomere attrition. I don't want anybody jumping on my back and saying that doesn't necessarily mean it's really aging and accelerated aging because it's just telomere attrition. Well, okay, I get that. It's one type of model here. So we're just comparing these two. And now we're comparing other types of damage that also accrue over time in both the in different rates. I think this goes a little bit faster. So now we're looking at damage, right? So they've established that in both aged mice and telomere attrition mice, obviously you get telomere attrition. And as a result, or maybe as a correlation here, you're also getting at the same time, more collagen produced, less of the types of cells that you expect to promote proper alveolar function, such as these AP2 cells and more of these collagen-producing cells. And you also see more indications of DNA damage. So figure 3 shows, again, same layout, looking basically microscopic analysis and then a histogram correlating to what you're seeing on the left. Induced telomere shortening and natural aging increase DNA damage, apoptosis, and macrophage senescence in the mouse lung. Okay, so more interesting things. So this is staining for DNA damage, and down here, it's gamma H2AX. So basically, it's a phosphorylated histone that's indicative of damaged DNA. Apoptosis marker, I think it's caspase. Yeah, caspase 3. Then you have P16, which is indicative of cells, undergoing senescence, P16. And macrophages, that's, so the marker for macrophages is F480 and P16 as well. So they're looking at macrophages as well, not just the epithelial cells in the lung, right? So that's the qualitative data on the left. Again, same thing, 5 weeks, 8 weeks. So a lot of work here, they really, again, I like that they used, these mice are all from the same background. So, what you don't want to be doing is comparing mice from different genetic backgrounds or, different litters. And, so right here, it's, these mice are pretty inbred and they're from the same litter. So whatever differences there are, you're trying, they're really controlling for all of the variables here. So I'm going to stop sharing here, and that's something that you can do. So I'm just going to say that it's something you can do with mice and something you can do with very similar genetic backgrounds that you can't really do, obviously, with humans. And that's what makes clinical trials really hard to interpret when you have a very small n or even a modest n, because people are very heterogeneous. So unless all of your people are identical twins, it's you're going to have error bars all over the place because different environments, different genetic backgrounds, so on and so forth. And these mice, bred in the same box, same environment, and pretty much as genetically identical as they can possibly be, except for the mutation that you see with the double minus TERT mutation, and then their wild type cohorts, which are as kind of genetically similar as you can possibly make it for a mammalian organism. Right? So that's what scientists like to do, is kind of control for all the variables so nothing else kind of rears its ugly head after you've done your work. So, okay. So clearly mice are not humans, and we all know the issues here. Mice have very long telomeres. Their rate of attrition, though, is a bit faster, and they live a lot shorter. But that's as darn good as we can get using a mammalian model right now with current technology. So we'll go back to sharing screen. Okay. So again, really good data here, really clean for what they have. Alveolar gamma H2AX, which is indicative of DNA damage I keep forgetting what that acronym stands for. I know F is for field, high magnification field, for what P stands for, but basically it's the amount of cells in your field of view, field of vision in a microscope, and you count them. Maybe by eye, but sometimes by software, and basically you score it. And the cells that got stained, you could see on the left side, they got little arrows that point out what they're looking for, right? So there's a little brown stained cell here. That's what's immunostained, and that's what they're measuring. So the numbers are higher to begin with your G3 TERT minus cells. So, they already have really short telomeres. So these your kind of, which I'm going to call your positive control cells, are already screwed up from the get-go. But you can see that these effects go higher. These, as the cells get older, the normal cells, the normal mice, right, from the same litter, the numbers go up as well, right? Basically equaling at very old ages, equaling what the screwed up mice, G3 minus mice already exhibit early on. And you see a similar effect with increase in caspase over time, right? So the trend is there. So basically, I'm going to scroll to next slide. So the trends are there. Basically, you have more damage accumulating as these cells are getting older. So we're not yet there where we're fixing these mice with, so what are we looking at here? So proliferation, right? So as cells, these are dividing cells that you're looking at, and as the cells get older and older, they divide slower and slower. So these are proliferating cells. So they start senescing basically, and that's one of the character hallmarks of senescence for proliferating cells is, well, non-proliferation. So you get premature telomere shortening. We've seen that, natural aging, and they reduce the proliferation of AT2 cells, macrophages, and club cells, and increase the differentiation of club cells. So I believe they use SOX2 as a marker. So as these cells differentiate, they stop proliferating. So I'm not going to go through this whole thing here, but basically, similar layout. You're doing microscopic analysis on the left, and the arrows point to what the researchers are looking for, and then all of the histograms are the quantification of all of this stuff, right? So the Ki67. So if we look at for example, so SOX2 is a differentiation marker. Ki67 is a proliferation marker. That's upregulated in proliferating cells, and you should see less cells in the field of view. So these are SF. So we'll just take one of these histograms as an example. Take the first one, SFDPC positive, which are the AT2 cells that are basically secreting surfactant, and looking at Ki67, which is a proliferation marker, and TERT++ cells. It's the white histogram, N equals 4 mice, and then N equals 4 G3 TERT minus mice. So you could see proliferation has already crashed for these G3 minus mice, and the proliferation also drops quite dramatically, as the mice get really old, for your wild type mice. So basically, you can go through the rest of that data, but that's also reflected in macrophages and club cells, which are another alveolar cell. And I believe what they're looking at here they're also showing, they're doing measurements, they're doing messenger RNA levels of telomerase, and they're showing that those levels go down as well. Okay, so I'm going to stop sharing here. Anybody has any questions? So basically, up to this point, the authors are setting the stage and showing what's going wrong with these mice, and they're comparing basically your normal wild-type litter mates that are TERT++ with your kind of hyper telomere attrition mice from the same litter that are being compared. And you can see that they have similar problems or manifestations of sort of accelerated aging and other effects such as increased senescence, shortened telomeres, increased expression of collagen and other factors, and lower amount of functional cells that are basically required for proper lung functioning. So the next slide I might skip over because we'll take a look at the histogram, and this is the reason is I'm not familiar with the technology. It's basically using a refractometer to look at. So basically, I mentioned that alveolar cells produce a surfactant, and basically that prevents your alveoli, which are these little sacs, from collapsing, right? So if you basically secrete something that's hydrophobic or like a detergent, right, it's going to prevent hydrogen bonds from forming and preventing things from sticking, right? So water has a lot of surface tension, a lot of electrostatic forces, and if you get water in your lungs, they're going to collapse. So your lungs are producing surfactant because there's a lot of humidity there to keep things basically open so they don't collapse, right? And those proteins that are part of the surfactant, you can actually measure them using this technology called I think a bubble refractometer, which basically uses glass and somehow measures the angle of bubble formation to basically show how much of this surfactant is actually there. So the concentration of surfactant, right? So basically, we'll go back to, and I admit I'm hand waving there because I've never used a bubble surfactometer before. And unless you work in a lung lab, you probably haven't either. So this is the data here. So this is a measurement that basically I'm going to leave up there for anybody who's more capable to basically interpret the bubble surfactometer data here. But basically just jumping to the figure legend, the measurements that they've taken from the surfactometer basically says that there's impaired surfactant activity, which is partially restored by telomerase gene therapy. So, so I don't know how to basically interpret the surfactometer results because I've not used this device here. Is my 2 cents on this. So basically, it's you're injecting it into a device that measures how much of this chemical is in that. So I might come back to this later. Basically, it's measuring surface tension of bubbles in this device or this machine. But at this point, they're adding in telomerase to these mice. So this is, oops, sorry about that. So this is using an adenoviral vector, AAV9. So basically, they also have a control, which is AAV9 empty, so no telomerase, and then AAV9 TERT. And they add this back into, I believe, both I think they're adding this, to, well, they're definitely adding it back to minus at 27 to 30 weeks. And I think they're adding it in 0, which is adding in extra copies to, if I'm not mistaken, the wild type mice. So now they're basically showing the same data that we've seen before, but looking at it from the angle of having extra telomerase put in, right? So let's scroll down to the kind of the figure legend. So according to this, telomerase gene therapy in the mouse lung results in telomere elongation and increased number of AT2 and club cells. So that's the protocol. So we have here AAV9 empty, AAV9 TERT. Scroll down over here. So they're looking at time weeks. So I'm trying to take a look at here, TERT plus, 21. So sacrifice and collection of lungs is at 21 weeks. I'm gonna just scroll down back to the other figures here to see how that looked for 21 weeks when it's 8 to 9, 42 to 49 weeks. So they didn't have that for 21 weeks here. Okay. So let's see. So they're looking at first telomerase levels. So telomerase levels AAV9 empty obviously is down. Telomerase levels go up. Mean telomere spot intensity. So AAV9 empty. Let me look here. So sorry, I have to familiarize myself with this chart here. It's been a while since I read that. Mean telomere spot intensity in club cells. Let's see, is that going up? Let's see here. Sorry, my apologies here. A little moment here because it's been a week since I looked at this particular figure here. So we're looking at the mean telomere spot intensity in the club cells, AAV9 empty. Yeah, okay. I was just got a little confused here because I was looking at empty versus TERT. I was just looking at that as from a different angle here. So let's see. Okay. All right, that's where I was confused. So I was looking at F here. So we're looking at percentage of short telomeres in SCGB1A1 club cells, and the percentage of, I was looking at long. I was like, wait, is that long telomeres? Why is that going down? No, it's short telomeres. So cells with short telomeres, the number goes down. Mean telomere spot intensity goes up. Okay, in SGB1A1. Okay, so that's why I was confused. I was like, they're looking at short telomeres, and the number was going down, which is inversely correlated. So that makes sense. TERT levels go up, obviously, if you express it. Mean telomere spot intensity goes up if we compare from empty to TERT. So the spot intensity goes up. So number of telomeres. So basically, you're seeing an increase in telomere spot intensity, increase in telomere length when you add in the vectors. And are we seeing now a modest increase in SFTPC-AT2 cells with TERT? Oops, got a chat question here. So the question is, do you understand why the authors focus their analysis on alveolar type 2 cells and club cells? Yeah, there's other cell types in the lung. They're focusing on these, from my understanding, is that these are, I think, the cells that are most affected in idiopathic pulmonary, fibrosis, and they're responsible for producing surfactant. So they're critical for lung function. Obviously, there's other epithelial cells there, and obviously there's also normal fibroblasts that are producing collagen for bronchi, but that's the reason that they're looking at these particular cells. Yeah. So so the numbers are modest here. I'm looking at the number of, so I'm looking at G and H. So SGB1A1, so that's the marker for club cells, and SFTPC for AT2 cells in the field of view. And after TERT activity, the cell number goes up. So these are, so, actually, the cell number goes up. This is with 21 weeks, I believe, which wasn't looked at in the other, So Steve says loss of this surfactant supports formation of fibrotic tissue. Yeah. So those of you who read this paper, you might want to, if you want to chime in on the 21 weeks time point, why that's why they chose that rather than a later time point. I'm not sure. Actually, what did it say here? 27 to 30. Oh, sorry about that. Go back to the figure. Sorry about that. Yeah, so they're actually much older. So that's why I was confused. They're 27 to 30-week-old mice, and they were sacrificed 21 weeks later for lung tissue harvesting. All right, so they're actually, what is it? What's my math? 30 weeks, 21, 51 weeks. So that's
pretty old for a mouse.
Yeah, sorry about that. I thought they were 21 weeks old. My mistake. So if I'm scrolling back here, yeah, so they're way up into the late middle age territory here that they have there. Okay. That's why I was a little confused there. I was like, wait a minute, why is it 21 weeks? All right. Okay, so good. Now we're at the discussion, but not quite yet. We've got three more figures, I think. So basically what we're seeing for these next figures is, improved lung functionality. So basically that's the same data that you've seen from the first 4 figures that I showed, but now with the addition of your AAV9, introduced telomerase subunit. So figure 7 is again looking at lung function from the angle of increased fibroblast activity, which you don't want to see. So here they say AAV9 treatment reduces fibroblast presence and activation and collagen deposition in the mouse lung. So same type of staining, on the left, and then quantification on the right. And I believe that this is also going to be 21 weeks after introduction. I don't see that in there. I think that's also 21 weeks after. So these are probably 50-week-old mice. So AAV9 empty, so TERT++ are the, aged mice, and then G3 TERT minus are the, your messed up mice with the short telomeres, and staining with vimentin and SMA, which are indicative of fibroblast activity, and serious red and mass-on trichome staining, which stains for extracellular matrix, particularly collagen. And then let's take a little. Sorry, I muted myself accidentally. That's what I get for using my finger to move things around. I have a touch screen here. So vimentin area, you would expect it to decrease if their hypothesis is correct, and you do see a decrease in vimentin area in the field of view. So both for your aged, I believe, roughly 50-week-old mice that have had the AAV9 vector introduced, extra copy with AAV9 TERT mice, and kind of similar effects across the board that you see when they look at, well, let's look at something else here. Let's jump down to E, peribronchial collagen, right? So you could see that number has dropped, so percentage has dropped from 6% down to 3%, so 50% reduction, right, in collagen. So that's good. And now we look at DNA damage markers, or basically markers that are indicative of cells that are responding to damage, such as apoptosis, senescence markers like P16, and DNA damage directly, such as MH2AX. So same figure, just with the addition of telomerase, your subunit. And if we look at alveolar gamma H2AX, which is quantification of the of the two boxes that you see in the upper left-hand corner for TERT++ and G3 TERT-, AAV9, and then right beneath it, AAV9 TERT, which is the addition of telomerase catalytic subunit in your TERT++ background and your TERT-, background G3. You can see that the gamma H2AX, which is indicative of DNA damage accumulation, goes down. Again, looks, say roughly 50%. And other markers also go down. So basically caspase, which is indicative of, apoptotic cells, and P16 indicative of senescent cells. So your senescent cell load goes down as well, which is pretty cool, because that's kind of that was the, I'm gonna stop sharing here. That was basically what they were going after in a clinical trial paper in humans with idiopathic pulmonary fibrosis, as if I recall, that they were targeting senescent cells directly. So they were getting rid of those cells. And here you see a senescent cell load go down just with the addition of extra telomerase. So that's, I mention that because that bodes well to me to kind of start edging forward with clinical trials. Because it was, well, they needed to run those a little bit more samples, because I think it was like 9 humans that were in that trial. So the safety profile was excellent. So removing senescent cells using senolytics was pretty safe. But they seemed to, there was some evidence in that it was improving functionality, and I think that work is being followed up at the Mayo Clinic with more participants. So if targeting senescent cells seems to be kind of the main way to tackle idiopathic pulmonary fibrosis and other chronic ailments of the lung that result from the aging process, then seeing a reduced senescent cell load here by adding in telomerase also ties into that and bodes well for support that this should potentially be a clinical trial for humans. So just making a note of that.
Oliver, it could also be indicative that the immune cells are also clearing house again in the lungs. So either the telomerase is either getting rid of senescent cells, or best guess, it's only a guess, that immune cells are actually starting to clear house again efficiently. So, but
yeah. Yes.
Biology's like that, isn't it? There's more than one way to skin a cat, as they say. It makes you think that telomerase and senolytics may be a good co-therapy. Get rid of, purge the cells, rejuvenate the cells.
Could be, yeah. So, I mean, it's certainly all of this data coming in is bodes well for the tackling aging by tackling various hallmarks of aging, and tackling them together might give an additive or synergistic effect.
Yeah, I mean, telomerase is very much a sort of divisive subject, isn't it? Really, there's a lot of people that think it's not going to help, and then the other half who think it is, and some people who think that telomeres are the holy grail. It's not really, but I think it could be useful. I definitely think it could be useful for things like fibrosis.
I'll even go a little further and say the fact that this is working in mice, which have extremely long telomeres, and therefore are, you're adding in extra telomerase, and you're improving telomere function in mice, and you're having these effects on an IDF model, bodes even better for a human application, because we have shorter telomeres. So, that's might be a stretch, but it's mice, it's humans, but that's the model that we have, and I think so far it's very promising. Let's put it that way. I could certainly see this not working in mice, and people saying, well, the mice, the telomeres are longer and they've got other issues, and maybe it'll work in humans, but it's working in mice. So, let's go back to, okay. So damage response is abrogated. Now we're going into a proliferation response. And I believe what we'll see here is increased proliferation. And what jumped out at me as I just immediately looked at B, which is your SFTPC, your quantification of what's on your left, which is your proliferating AT2 cells, and AAV9 empty. The numbers are pretty low, proliferating, and are pretty high for post telomerase treatment for both wild type and telomerase negative. So you have more proliferation, and also in macrophages as well, right? So that's your immune cells as well, club cells as well. So I think I'm gonna skip through all of this stuff here. It's messenger RNA levels of telomerase. So figure 9 is treatment with AAV9 TERT restores the proliferation of AT2 cells, macrophage, and club cells, and reduces the differentiation of club cells, which was the SOX2 marker, right? So more proliferation taking place, and less senescence, obviously. Was that it? I thought we had another figure here. That's figure, that's figure 9. Did I skip a figure? Well, I did skip figure 5 because that was our surfactant analysis, where the cells, they basically, not familiar with using a bubble surfactometer, but suffice it to say, the cells are, producing more surfactant, and according to that measurement, that device that they were using, that goes down, which I believe. All right, wow, okay, I thought there was, did I skip figure 8? No, I didn't skip figure 8. I don't think that was it. Yeah, okay. So the one comment that I'm gonna say is that mice do have very long telomeres, although the telomere attrition, I think, is much faster. I don't have the rates in front of me. The one thing, the one comment I could make is, there's correlation and causation, two different things. It certainly suggests that, telomeres, which are attrition is 100 times faster, according to Steve. The telomeres are the stretches of DNA along with the proteins that basically cover them, at the ends of chromosomes, and they vary in length from different organism to different organism, and of course they're there to basically, I think Elizabeth Blackburn referred to them as aglets. If those of you who don't know what an aglet is, I had to look that up. That's basically the little thing at the end of your shoelace that prevents it from fraying. And the proteins and the telomeres prevent the fraying of chromosomes by basically attracting an enzyme, telomerase, that basically is a reverse transcriptase that basically adds extra bits of information at the end, repetitive sequences. The telomerase itself might actually be doing other functions somewhere else as well. So, it's not implausible that it's not just the telomere length themselves that's basically, causing a reversal of all of these effects here, but it might be the TERT itself. The only way that you can really clear that up, I think, is by using a mutant TERT that basically lacks catalytic function, but has other functions. So I'm going to toss that out there. Yep, Steve says TPE also influences gene expression, definitely, but that would be through the, the extension of telomere. As the telomeres get shorter, right, different factors leave and they do other things in the cell. It could be TPE, telomere position effect. It could be TERT itself having other effects somewhere else, right? So, so just kind of there's room here, despite all of the kind of really detailed work that the authors here did, showing that, basically you get a reversal of all of these effects in a mouse model of IDF, or where's IPF, idiopathic pulmonary fibrosis, a reversal of all these effects by adding in catalytic subunit of TERT. And as a consequence, of course, you get an extension of telomeres. But is this solely due to an extension of telomeres, or is there an additional effect? I would kind of posit from the TERT as well, doing something in other locations.
Well, TERT also interacts with PCG1A, which is also, I think it was it DePino who worked out there was an axis between telomeres, mitochondria, and P53 via PCG1A. That's a good paper. It's quite a bit old now, but it does go to show that TERT also is important in the balance of PCG1A, and therefore by proxy also mitochondrial count and function. Good paper, actually. I think it was 2012. I think it was Ron DePino, I think. I'll dig it up.
I have a vague recollection of reading that. But yeah, so that's my comment there. Certainly not a strike against the paper. More of a statement that there's more things to tease out here. Certainly the effects themselves are what we see here. They're not gonna change. But I would be interested to see how much of those are effects due to telomere elongation. As a scientist, maybe that's not so important from a clinical standpoint. If you're going to be running into clinical trials, this is more important from a basic research standpoint to kind of tease out the effects of what TERT is doing. If there's anything outside of just telomere elongation per se, to basically reverse these effects that were seen in this paper. Okay, do we have any comments on this work here?
I mean, my big question is what cell types are their effects being mediated by? Because their AAV9 construct, I think, targets like everything. It's an IV injection, so everything in that mouse is going to get the transgene. They got some evidence in figure 6, like their panels C through G, showing that like they probably had telomerase activity going up in their two cell types of interest. But that doesn't necessarily mean those are the cells that are mediating the effects. It could be something they're not looking at. Yeah,
I mean that's a good observation. AAV9 is not. They're not targeting. This was done. I think intravascularly, right? They injected this into mice. They didn't aerosolize it and put it directly into the lungs. And AAV9 is pretty much going to go everywhere. So you would think that it would be, that these effects are going to be directly, working on lung function. But obviously, if it's going everywhere, and as Steve mentioned, if you improve, here in the lungs, the macrophage functionality is improved. If you're getting better clearance of senescent cells through an enhanced immune response, perhaps the immune system is also getting improved through the AAV9 TERT, right? So it might be targeting immune cells. They didn't look at that, right? So that's another interesting observation. So if there's a follow-up paper, maybe there's enhancements in other cell activity that translates to improved lung functionality, such as basically maybe an enhanced immune function. So that's a good call. So thanks for bringing that up.
I feel like it's kind of an important issue if they're thinking about like potential therapies, because like when I think about telomerase, the big caveat is you might be able to make cells last longer, but you'll probably also increase cancer incidence because cells can divide more often. And I'm guessing that the cell of origin for most lung cancers probably isn't type 2 alveolar cells or the club cells. Like maybe it's okay to increase telomerase in those cells. And maybe there's some other cell that if you increase telomerase, you get a cancer risk. It'd be cool if there were some viruses of tropism for the cells that actually make surfactant. I don't think that virus exists right now, but I feel like if I was in this field, that's what I'd be thinking about right now.
Yeah. I mean, that's a good point. I think what we'd see here in, like, I'm just kind of projecting kind of like for clinical trials, people who have this condition, you know, whether or not they get the AAV9 vector in a clinical trial through the bloodstream, or whether it's, you know, through some sort of aerosolizable method, you know, which might be lower risk because it's, you know, you're localizing the targeting of your telomerase and minimizing it getting elsewhere. I think probably for patients that are suffering from this disease, you know, the potential of an increased cancer risk might be waived, just because I think, according to this paper, you have like a, you know, if you're at a certain point of diagnosis here, you have a lifespan of maybe 4 years to live with this disease. So it's a pretty aggressive disease. So, so, you know, the cancer risk might be, you know, cancer risk in 8 years might be kind of moot at that point, you know, because it's because of you dying from IDF. But for other individuals, you know, such as the normal aging, through the normal aging process, yeah, you would probably want more rigorous experiments done, more, you know, controls to make sure that you're not getting AAV9 elsewhere. Although potentially targeting telomerase to other cells might also be beneficial to the normal aging process as well. So you might want to target many other cells. So, so I don't know if AAV9 is, so I guess the bottom line is, I don't know if that particular vector is the best vector to use, although it could be. I think it's the best one that we have right now.
One that Blasco uses all the time.
Yeah.
She's been using it since what, 2011, when they first did the original telomerase therapies.
Yeah.
It's always a favorite one. So I don't know. It's interesting though, isn't it? But yeah, Jedlie on Facebook has noted that the catalytic subunit itself, TERT, migrates to the mitochondria and helps to manage ROS as well. Okay. So again, it's going to be a reduction of ROS, and, you know, therefore local environmental inflammation, which again, a reduction of inflammation could spur the immune system to start working again properly.
Yeah, I mean, it could definitely be two effects here. I mean, we could, it could be, some of the effects that are noted in this paper could be through the extension of telomeres, and some of them could be additional effects that Kurt is doing in other cell types and, you know, subcellular organelles such as mitochondria. They haven't looked at that in this paper, so which is great for researchers because you've got all of that to look at now.
So yeah, I definitely think there's something in this. I mean, I can't help but just recall last year's paper when you met Maria at 2019, mm-hmm. And they just done that study about telomere attrition, and they found it was the rate of the burn down that was sort of supportive of how long something probably would live. So that's interesting, really. Telomeres have clearly got an epigenetic and genomic stability role. So I think they're more important than some people think they are, but I think they're more important for some reasons that some people don't realize they are. It's not just about how long they are. Mm-hmm. And having super long telomeres as a human is not a good idea. You need what you need. So, because I'm pretty sure they've actually made super long telomere mice, and it didn't go well.
Yeah, well, you got to realize that there's a, well, I mean, there's a balance to all these things. Telomeres titrate factors, right? Telomere position effect. So if you're binding factors and pulling them off from other regions, then that's not going to be good as well. So, you know, things are in a balance, to put it in a very new agey way, right? It's gotta be balanced, man.
Gotta be Pappins. Oh, good grief! But yeah, I honestly think Blasco and the CNIO, they're step by step getting closer to the day where I think we'll see it tested. I think we'll see a human trial. I don't know what she said last year. Maybe within the next 5 years. I think she was fairly open to that. 5, maybe 10 years. I think probably 5 years.
Well, certainly this paper shows great promise. Again, using this as using an illness such as IDF, where it's I think it's a pretty appalling thing to live with, and there's really no good treatments, and you're going to die in a very short period of time if you don't get any sort of cure. I would think and hope that would certainly accelerate the process of clinical trials in this case, especially with something that appears to be so promising. I'm just speaking for myself. If I had that illness, reading this paper and reading, Kirkland's work, sign me up for an aerosolizable AAVS9 treatment coupled with senolytics.
Well, I mean, there's absolutely no current, treatment for it. So I understand that in some cases where there's literally no other alternative or current, drug or whatnot, it can sometimes accelerate the process. So there you go. Fingers crossed. I think, honestly, I've said it before and I'll say it again. I think if anybody is going to deliver telomerase gene therapy in a safe and, proper way, then it's going to be CNIO and in particular Maria Blasco. It's her thing. She's been at it for over a decade, but honestly, I think she's the person that's going to deliver it. There you go. I mean, that's I'm not sponsored by her or anything. I'm just a huge fan.
I don't think anybody's sponsored by a research lab. I think that goes the other way.
Yeah, no, I'm just a gigant. I think it's eventually going to pay off, but she'll do it when it's safe to do it, and she's being very sensible about it, which is good because you wouldn't want to run off somewhere and try things that are not really safe, would you? Or might not be safe, because that would be dangerous.
All right. So, do we have any more comments? Any great questions thus far, Michael? Those are good ones.
I don't want to drown you in questions, so I'll make this my last one. This is more of a technical thing. Figure 2, first column near the bottom, they're showing the progression of their pulmonary fibrosis in their mouse. And I've actually never seen sections for pulmonary fibrosis before. And I'm trying to figure out what's going on. So clearly there's much smaller, what's the word, vacuoles, alveoli. There's smaller air sacs as age progresses. But what's going on with the tissue? Is there proliferation of cells, which is leading to smaller air sacs, or are the cells just getting wider? Is it bigger cells or more cells? I'm wondering what's driving the loss of air sacs.
Cells do. They found out last year, didn't they, Oliver? It was last year. That cells, or senescent cells in particular, or as cells approach senescence, they do tend to get larger.
Yeah. I don't know. It can be a combination of the two. It could be enlargement and also attrition. Certainly you get attrition of cells, and the cells that are left over are less functional. There must be a good review paper out there that covers kind of cellular activity in general as cells age, because those are just some generalizations I just mentioned, but I mean, looking at it from a kind of histological, physiological perspective, right? As tissues age, right? There must be, I'm just thinking that it's got to be some really old school studies that there must be a compendium of of what happens to tissues and what happens morphologically, and in I guess other manifestations to cells in different organ types as humans get older and age, right? I would think that I would be surprised if there's not like a really weighty kind of geriatric tome that covers that somewhere.
You just need to crack open a pathology textbook.
Yeah.
But like their therapy. So Steve mentioned that what the cells have in common is that these are surfactant secreting cells, and that makes sense. So you might expect that since they see a whole lot of beneficial effects via their intervention, that it's mediated by more surfactant. So the air sacs can just inflate better. Mm-hmm. So like I'm kind of wondering if when they are looking at their idiopathic age-associated pulmonary fibrosis, is this just like a partially deflated lung? Or is it something else? Is it like cellular proliferation or like cells are just bigger because they're senescent? Yeah. This is maybe trivia, but it's the question it's in my mind anyway.
You mean what out of all these things that we just mentioned, what's the most contributing factor to the pathology of the disease itself?
Yeah. Is it like with age, do you have like fibroblasts proliferating? Because in like smoking pulmonary fibrosis, it's considered scar tissue that accumulates.
Yeah.
Or is it cells are no longer senescent, so they're just bigger and there's less room for air sacs? Or is it there's less surfactant, so all the air sacs shrink?
Certainly my recollection from the paper we went over with from the Kirkland lab, they were looking at fibrosis, right? They were looking specifically at, that was kind of mentioned repeatedly through the paper, that you basically had kind of a transition to more kind of fibrotic tissue. And having more fibrotic tissue, obviously that tissue itself is going to be less gas permeable, and it's going to be, primarily less gas permeable. So it's, so then if that's the case, then, not having enough surfactant might be a small part. I don't know what the percent. So what I'm thinking here, maybe you're kind of thinking the same thing. Correct me if I'm wrong, but what percentages of the pathologies come from the different aspects of this disease, right? So if you have less alveolar cells are producing surfactant, how much improvement do you get by just adding in, let's say, more artificial surfactant, right? Maybe 10%, 20%. But then if you also have fibrotic tissue there, adding in more surfactant isn't going to improve it even further, because those cells are basically not as elastic, not as gas permeable. So having more surfactant you're still going to have a reduced lung function. You're not going to get to 100%. So I don't know. Were you thinking along those lines?
Yeah, I think we're on the same wavelength. I think that's what I'm wondering here.
Okay. Yeah, I'm wondering the same thing too. I don't know. So it could be, so it could be that targeting something like telomerase would get to the root cause of all of this and just basically get your lung functionality up to a theoretical 100%. Or maybe, there could be other interim therapeutics that basically can target certain aspects of it that can, like putting back more surfactant, for example, but that only gets you to a certain level, assuming your idiopathic pulmonary fibrosis isn't so advanced that, most of your lung is just fibrotic tissue. So, yeah.
Thanks.
So, yeah, I think that question, if we had a specialist on board, a lung surgeon, I think that would be the type of question that we would direct towards that specialist. But there might be a review paper on that. But I'm just guessing that might be the case, but I'm not the expert on it. So yeah. Okay. So I think that concludes our journal club. So a little bit past the hour here. Went over pretty much every figure except for FOG, which was a surface bubble refractometer, which measures their surfactant ability of the cells. I have to skip over that because I'm not well versed in that piece of tool. But suffice it to say, it seemed to improve the surfactant activity. And, the results in this paper suggest that you had more of the cells that produce surfactant and less collagen production. So hence potentially less fibrosis in these cells. So it's very promising. Maybe it would, it certainly supports, the hallmarks of aging. And I'm looking forward to seeing, this being introduced as a potential therapy for people that are suffering from this illness. And, and obviously the end goal is then to see if this is applicable to people that are very aged and also people that are not so very aged, to basically improve lung functionality. And clearly in those cases, you want safety to be of the utmost. So, anyway, Steve, I guess that's if we have no more questions, I think we're wrapping up here.
It seems a good juncture to do so, Oliver. Yeah, and there's an absolute tornado going on outside.
Oh, really?
There's some
I could see trees swaying in the background.
Yeah, I think I'm gonna have to go and check on that because I've just heard some crashing. So yeah, on that note, we don't want any more storms, guys. So stop sending them across the Atlantic, please. The UK is fed up with storms. And on that note, thanks for everyone who joined us. Hope you found it interesting. I personally think it's exciting. Looking forward to seeing a human trial. Don't know when that'll be. And thanks to the heroes, especially who joined us today. And we will be in touch regards the next topic. There's so much going on. Who knows what it could be. And we'll see where we go from them. So thanks to the heroes who have made this show possible. And if you're interested in supporting this and the many other wonderful things that we do, you can check us out at lifespan/heroes and see if that's something that you might like to get involved with. So until then, we will see you again. We'll see you around about the same time.
Yep. All right, guys and gals. Take care.
Later.